Hey everyone! Today we are diving into one of the most fascinating areas of genetics, modifier genes. This exciting topic is brought to you by the Shah sisters, Amna and Farah, from the MHSc in Medical Genomics program at the University of Toronto. In this webinar, we'll explore how modifier genes impact genetic traits and why they complicate classic Mendelian inheritance patterns. We will break down the concept of various modifier effects, including penetrance, expressivity, dominance modification, and pleiotropy. We'll look into the mouse model example where mice with single gene mutations are bred with different genetic backgrounds. And finally, we'll have a few quiz questions to test your knowledge. Let's dive deeper into the background. The consistent expression of Mendelian traits across generations is actually more of an exception than a rule, both in mice and in humans. Why? Well, it's largely due to genetic modification. So, what is genetic modification? Simply put, it's when the expression of one gene influences or alters the expression of another gene, resulting in a wide range of phenotypic variations, which we refer to as modifier effects. Now, you might wonder that what causes the variable phenotypes seen with the modifier effects? Well, it's a mix of alternative alleles, environmental factors, and importantly, modifier genes. Allelic and environmental variations are well known and documented, but in certain cases, even when these factors are accounted for, we still observe phenotypic diversity. This is where modifier genes come in. Let's start with penetrance. Penetrance is about probabilities. It tells us how many people with the specific genetic change actually show the related trait or condition. Think of it this way. In simple Mendelian inheritance, if a gene has 100% penetrance, everyone who carries the mutation will show the expected trait. But genetics isn't always so straightforward. Often we encounter reduced penetrance, where everyone who carries the mutation will not show the expected trait. This means people with the same mutation can be healthy or have the disease. When researchers study these unaffected individuals carrying the genetic mutation, they found clues pointing to another gene at play, which is a modifier gene in action. These modifier genes act as genetic influencers. They can enhance or suppress the effect of the main gene mutation, the DFNB26 gene on chromosome 4q31 affects non-syndromic deafness. While most people who are homozygous for the DFNB26 gene experience non-syndromic hearing loss, some individuals in a family where DFNB26 was mapped are homozygous for the gene but still have normal hearing. This is due to a dominant modifier on chromosome 7 identified by a recent study, which protects them from hearing loss and accounts for reduced penetrance. Next up, let's talk about expressivity. If penetrance is about whether a trait shows up, then expressivity is about how that trait actually manifests, its severity or mildness in any given individual. Expressivity helps us understand the range of a phenotype associated with a particular genotype. Modifier genes may enhance or reduce the expression of disease-causing genes by altering how strongly certain traits appear. They can make a mutation more noticeable, leading to severe symptoms and result in milder symptoms or even make it resemble a normal wild-type version of the trait. What's critical to remember is that expressivity is only relevant in cases where the trait is already penetrant, meaning it shows up in some form. To understand this further, let's take a look at an example. Familial hypercholesterolemia, FH, is an autosomal dominant trait impacting about 1 in every 500 people. It's a condition where individuals have significantly elevated cholesterol levels, putting them at a much higher risk for cardiovascular disease. FH heterozygotes are predisposed to early cardiovascular disease, while homozygotes can develop life-threatening heart conditions by their 20s. For instance, some people with familial hypercholesterolemia have up to 25% lower bad cholesterol (LDL) levels than expected. This suggests a cholesterol-lowering modifier gene. Surprisingly, it not only lowers LDL in people with FH but also in those without the condition. This example highlights the relevance of expressivity. The LDL lowering gene doesn't eliminate FH, but it alters the severity of cholesterol levels in individuals with the condition.
Let's dive into the concept of dominance modification. Generally, we think of dominance as a clear-cut relationship, where one allele of a gene suppresses the expression of another allele at the same locus, determining the phenotype in a heterozygous individual. However, modifier genes can blur this distinction. They can make a trait that is dominant in one context become semi-dominant or recessive in another. Just a refresher, semi-dominant means where the mutation of one allele leads to an intermediate phenotype, but the disease phenotype may be less severe. And recessive means where both alleles needs to be mutated to show the disease phenotype. This suggests that genetic modifiers can shift how dominance is expressed across different individuals. Let's explore an example of dominance modification in a condition called retinitis pigmentosa, a genetic disorder that causes progressive vision loss. In the autosomal recessive form of retinitis pigmentosa, the condition is caused by homozygous mutations in the PRP1 gene. This means that individuals with two mutated copies of PRP1 gene develop retinitis pigmentosa. In the heterozygous state, where there is one mutated PRP1 allele and one normal allele, the individual maintains normal vision because the normal copy fully compensates for the defective one. However, things become a bit more complicated when a modifier gene called ROM1 or ROM1 interacts with the PRP1 gene. In this case, one mutated copy of PRP1 and one mutated copy of ROM1 together creates a double heterozygous effect. This interaction between the modifier gene and the PRP1 influences the phenotype of the PRP1 gene, leading to the development of retinitis pigmentosa. Even though the PRP1 mutation alone might have been harmless in its heterozygous state. This example demonstrates how dominance modification caused by modifier genes can result in unexpected genetic outcomes such as progressive vision loss in retinitis pigmentosa. Alright, let's move on to pleiotropy. Pleiotropy occurs when a single gene mutation causes a variety of seemingly unrelated symptoms. In other words, one gene can influence multiple traits, even if they appear completely different. For example, a mutation might affect both skin color and lung function, which seem unrelated but are connected through the same genetic change. Now, this is where modifier genes come into play. Modifier genes can determine which of these pleiotropic traits will actually show up in an individual. This means that depending on the genetic background, a mutation might lead to one set of symptoms or an entirely different set, or it might even create a unique trait combination that only occurs in specific genetic contexts. This is possible because the modifier genes interact with the pleiotropic gene. Let's look at an example of pleiotropy in familial Mediterranean fever and how modifier genes come into play. Familial Mediterranean fever is an autosomal recessive disorder caused by a mutation in the MEFV gene. It leads to periodic fevers and episodes of painful inflammation of the serous membrane, such as the lining around the lungs and abdomen. A major complication of familial Mediterranean fever is renal amyloidosis, where abnormal proteins build up in the kidneys, potentially causing kidney failure. Interestingly, not all familial Mediterranean fever patients develop amyloidosis. This difference points to the influence of modifier genes. Recent research has shown that a polymorphism in the serum amyloid A gene or SAA1 gene influences the likelihood of developing amyloidosis in familial Mediterranean fever patients. So, while the MEFV mutation causes the core symptom of familial Mediterranean fever, a polymorphism in SAA1 gene acts as a genetic modifier, increasing or decreasing the risk of renal amyloidosis. Now, keeping the concept of genetic modifiers in mind,
let's look at the mouse model.
The wild-type mice in strain A have a homozygous genotype and display a normal coat color phenotype. Mice that are heterozygous for the M mutation carry the mutation but do not exhibit any coat color changes because the M mutation is a recessive condition. Therefore, only homozygous mutant mice show the variegated spotted coat color phenotype associated with this mutation. To investigate how this mutation behaves in a different genetic background, specifically strain C, scientists use a technique called backcrossing. In this process, they repeatedly mated homozygous mutant mice from strain A with wild-type mice from strain C, eventually creating a population in strain C that carries the M mutation in a new genetic context. In strain C, the following observations were made. Wild-type homozygous mice continue to exhibit the normal phenotype. Heterozygous mice are phenotypically affected, indicating that strain C mice exhibit a color change from orange to brown compared to strain A. This change in phenotypic expression represented a shift from a recessive trait to a semi-dominant trait. Consequently, this alteration is referred to as dominance modification. Furthermore, a change in body size was observed. Half of the mice maintained a normal body size, while the other half became obese. These variations in phenotype reflect a change in the expressivity of the trait. In homozygous mutant mice, there was also notable differences in body size compared to strain A. While all mice in strain A were lean, all mice in strain C were obese. Additionally, the coat color shifted from spotted brown in strain A to a solid brown color in strain C. These findings indicate that the M mutation leads to multiple trait alterations. As a result, both variable expressivity and pleiotropy were observed in the affected mice. In conclusion, the M mutation in strain C exhibits dominance modification, altered expressivity, and pleiotropy, demonstrating how genetic backgrounds can significantly influence the impact and severity of a mutation. Let's see if we have mastered the content with the quick challenge. Welcome to the final segment, Gene Genius. Question 1. In neurofibromatosis, individuals with the same mutation in the neurofibromin gene can show a range of symptoms, from mild skin spots to severe neurological problems. This variability in symptoms among individuals with the same genetic mutation is an example of option A, variable expressivity, option B, complete penetrance, option C, simple Mendelian inheritance. The correct option is option A, variable expressivity. This is because variable expressivity occurs when individuals with the same genotype display a wide range of symptoms in severity. In neurofibromatosis, this means some family members may have mild symptoms while others experience more severe complications. Question 2. In retinoblastoma, not all individuals with a mutation in the RB gene develop the disease. This phenomena, where some individuals with the same disease-causing mutation show symptoms while others do not, is an example of option A, variable expressivity, option B, incomplete penetrance, option C, complete penetrance. The correct answer is option B, incomplete penetrance. This is because incomplete penetrance occurs when not all individuals with a disease-causing allele express the related phenotype. In the case of retinoblastoma, some people who carry the RB mutation do not develop the disease, illustrating reduced or incomplete penetrance. Question 3. In cystic fibrosis, individuals with the same CFTR mutation can experience a range of symptoms, including lung infections, pancreatic insufficiency, and digestive issues. Some cystic fibrosis patients, however, have only mild lung symptoms and no pancreatic problems. Which genetic effect is likely responsible for this variation in symptoms? Option A, variable penetrance. Option B, modifier genes affecting pleiotropy. Option C, epistasis. The correct answer is option B, modifier genes affecting pleiotropy. This is because modifier genes can influence which specific symptoms of cystic fibrosis are expressed, demonstrating pleiotropy by causing a single cystic fibrosis mutation to lead to diverse effect across different organ systems. Question 4. Penetrance is the dash a trait appears, while expressivity is the dash of that trait. Modifier genes can influence both. Option A, likelihood, severity. Option B, severity, likelihood. Option C, frequency, intensity. The correct answer is option A, likelihood, severity. Because penetrance refers to the chance or likelihood that a genetic trait will appear, while expressivity refers to the severity or extent to which the trait is expressed. Modifier genes can influence both aspects by altering the likelihood of the trait's appearance and how strongly it presents. In summary, modifier genes are those genes that influence the effect of other genes. They don't cause traits themselves, but adjust how other genes express traits, leading to differences in how genetic conditions or characteristics show up in individuals. Penetrance is the likelihood that a person carrying a specific gene will actually show the associated trait or disease. Modifier genes can reduce or increase this likelihood. Expressivity describes the range or severity of symptoms in individuals with the same genetic mutation. In dominance modification, modifier genes can adjust how dominant or recessive a gene appears, sometimes making dominant trait less visible or making recessive traits more pronounced. Pleiotropy occurs when a single gene mutation causes multiple seemingly unrelated effects or symptoms. Thanks for watching. If you enjoyed the video, don't forget to like, subscribe, and comment below. See you next time.